Thank you, Lisa, very much. And welcome everyone as Vikela Earth celebrates Earth Day this week. And we've just heard a fascinating account of what happens when we go in and deliberately upset an ecosystem and the devastating effects that can have on, in this case, agriculture. And I'm going to talk to you today a little bit about another specific ecosystem, coral. And coral reefs provide critical habitat for species such as sea turtles, many fish, and form their own unique habitat. But we're struggling also with, with these habitats as well, with these ecosystems. I'm looking for my slides. Thank you so much. So if we talk about the coral crisis, some of you may be well aware of this. So coral reefs around the world are challenged by climate change because that's causing both acidification of the water, but it's also causing an increase in temperature. And what's happening is these environmental stresses then lead to coral bleaching, which will eventually kill the coral, and an increased susceptibility to disease. And so for those of us that work in the field of conservation, what we know is that the key thing is to try and preserve genetic diversity. If you imagine some disease comes through a population and the majority of the animals might succumb, but a few individuals might have a slightly different genetic adaptation, which will allow them to survive the impact. And so this is the same with coral. So there are many ways we can preserve these genes from coral, so we have the maximum genetic diversity preserved. And one way is through coral nurseries, like you can see here in this picture. But what we do at CSARC, we employ a different strategy. And what we're doing is we're cryopreserving sperm. And we do this for many species. And it's interesting, to learn that coral also have gametes, have sperm and eggs, like every other animal. So why do we want to freeze coral sperm? Well, we talked about how critical the role of coral habitat is for all kinds of different species. And what we're finding is with some of the genotypes where we've been able to go in and find out the genetic makeup of individual corals, we found that some are actually more disease resistant and some are a little bit more heat tolerant. So if we can bank genetics from these particular strains of coral, then we're starting to generate some insurance for the future. And then we can introduce them back into the population, if necessary, to sort of infuse the population with new genes. But we're also banking from many other different corals because we don't even know all the benefits their genetics may confer. And so to be safe, we're banking as much as we can from as many corals as we can to provide this insurance for the future. So if you've ever heard of a genome resource bank, sometimes it's nothing more fancy than what you see here. So this dewar is specially adapted to contain liquid nitrogen. And liquid nitrogen is so cold at minus 196 degrees Celsius that by the time the samples are preserved in there, they are metabolically inert and can be preserved for hundreds of years. And so sperm banking is a way of preserving genes for the future, providing this insurance to guard against catastrophic loss. And the genetic diversity preservation is a key thing if we're going to preserve species. So how do we collect sperm from a coral? Well, we work with staghorn coral, Acropora cervicornis. And what's interesting about this particular species is they spawn once a year after the full moon in August for only about 3 to 4 nights, and that's it. So our time window is very narrow to collect this incredibly valuable genetic material. And what happens is the divers go down and they put a net tent over the coral. And if you look at the photograph below, you can see little mustard seeds being released from the coral, and those are gamete bundles. Those contain both sperm and eggs, and they will float up to the surface. But what we can do is we can put a tube at the top of that net, and you'll see in that top left-hand photograph, we can catch those gamete bundles, and then we take them to the lab for processing. So those gamete bundles break down quite quickly, and that will liberate both sperm and eggs, which can be separated. And yes, if you look at the picture on the left, we're actually using a gravy separator to separate our gametes. So the sperm are in the cloudy portion below, and the eggs, which are buoyant, float up to the eggs. So we're able to separate the sperm from the eggs, and then we can treat the sperm and cryopreserve it. We haven't yet developed a technique for cryopreserving the eggs because they're relatively large and very lipid dense, which makes them extremely challenging for cryopreservation. So for those of you who've been wondering, what does a coral sperm look like? Here it is. It's a very classic sort of cell, something that might seem familiar with other species. And it's got a head, a midpiece, and a tail. And if we can, there's actually a video on the right. I don't know if we can click on that and play that video. Yep. And so this is what we see when we look under the microscope. Thousands of sperm cells all buzzing around super quickly. Can you play that one more time? It's a very short clip. And you'll see that the sperm are motile, and this is what we're looking for. But they have a very limited amount of time when they are motile. So it's important for us to get those gametes as soon as we can so we can get them cryopreserved before they effectively run out of energy. And so this is how we freeze coral sperm. You'll see on the left, we've got labeled vials. And so what's critical is that the vials are labeled with sufficient information so that 50, 100 years from now, somebody can go in and retrieve a vial and know exactly what's in there. And then we put the sperm in a solution with a cryoprotectant, and we freeze it above liquid nitrogen, which is what you see in the middle. And then we will clip those vials onto a cane, and then they get stored in that dewar. And the technique we use has been adapted from Hagedorn's paper in 2017. And this group are working with palmata, with elkhorn coral, but the protocol works the same for staghorn coral. This is just a little bit of data just to show that on the third night, we start to see a reduction in fertilization rates if we're doing in vitro fertilization, which drops off quite quickly. So if you look at the first point at 11:15, our divers are coming up with the gamete bundles and we're starting our fertilization experiments. But by 1 o'clock, that's only 2 hours later, we're seeing a dramatic decrease in fertilization, which means our sperm are now dying. So we only have about 1.5 hours if we want to bank the sperm to get the best results. And so what we're doing now is we're adapting our protocol and we're figuring out ways of actually freezing the sperm whilst we're on the boat. So as soon as the divers come up, they hand the samples to us and we're now processing them and trying to preserve them whilst we're on the boat so we don't have to wait for the boat to come back into the dock and then for us to get back to the lab. So time is of the essence in many, many ways. And this is one way how we're preserving coral sperm. This is just an example. Of course, the other important thing is making sure you've got a really clear record as to what you've got banked and where it's from. So we record the location, we record data about the motility of the sperm. And to date, we've banked over 1,000 vials from 17 different genotypes, and we are only one of several labs who are working to preserve coral sperm. So then we can use those sperm, we can thaw them, we can mix them with the eggs, and there's some pictures of embryos at the top as they go through their embryo development phase. That last stage at 10 to 12 is affectionately known as the cornflake stage or the prawn chip phase, because those are the later stage embryos. And then if we're lucky, they will settle and will eventually grow into individual corals that can then be outplanted onto the reef. So that's a very short account of one of the strategies we're using to help preserve genes, to preserve coral reefs for the future. And so our call to action, what can you do to help? Wear coral safe sunscreen, that's great. Turn off your lights when you don't need them to reduce energy consumption, and reduce, reuse, and recycle for a greener planet. We work with many scientists all around the US and the world who are all working on trying to preserve the staghorn coral, and the group is known as the Acropora Recovery Implementation Team, which just goes to show, it was mentioned earlier, the importance of collaboration and reaching out and partnering with as many individuals as possible to get to really maximize efforts. And this is something that Vikela Earth is really working hard on and really has a good network of these collaborators. Well, thank you for your attention, and feel free to put any questions in the chat.
Vikela Earth.